Hello, my name is Viviana Martins and I'm from the University of Minho in Portugal. The title of my presentation today is Exogenous calcium shifts the grape metabolic profile, promoting stilbenoid accumulation. As we know, grapevine productivity is affected by environmental challenges. And in particular, heavy rains before harvest cause fruit swelling, flavor dilution, and skin cracking. And this, of course, leads to major productivity losses. This problem of skin cracking is actually very common to other fruits, including sweet cherries and tomato. To overcome or minimize this problem, calcium supplements have been increasingly used to increase the firmness of the fruits and thus their resistance to environmental stress. And this is thought to be due to the multiple roles of calcium in plant cells, which includes structural roles in the cell wall and membranes. To understand if the application of calcium in field conditions could minimize the cracking of the skin of grape berries, we established field trials in which we selected the most prominent red and white varieties of the largest wine region in Portugal, the region of Minho. We then established two different treatments in which one set of vines were sprayed with a solution of 2% calcium chloride and a surfactant agent just to increase surface adhesion, and another set of vines were just sprayed with water and the surfactant agent, and this was used as a control. Both treatments were applied to the vineyards 3 times during fruit development. Our first goal was to confirm if calcium sprays could effectively increase grape berry firmness, and for this we established compression and perforation tests. In the first test, the test berries were placed between 2 metal plates and we measured the force necessary to crush the fruit. In the second test, we had a 1 millimeter diameter needle and we measured the force necessary to perforate the fruits. Results showed that the force necessary to crush and perforate the fruits was higher for calcium-treated fruits. And so we were able to confirm that our treatment was effectively increasing fruit firmness. The next step was to scan the surface of the skin of the fruits by electron microscopy. Results showed that the surface of control fruits was very rough. It had a significant extent of microcracks, and this of course favored the lodging of filamentous fungi. In contrast, the surface of calcium-treated fruits was very smooth. It had less microcracks at lower extension and also lower degree of colonization by filamentous fungi. Then we measure the cell wall pectin content, and for this we use the fluorescent dye named choryphosphine O, which specifically binds to pectins. Results show that control fruits display the mild fluorescence of this fluorophore, so they didn't have very high accumulation of pectin. However, in fruits treated with calcium, we saw a pattern with several patches of very strong accumulation of pectins. So this confirmed that calcium treatments were favoring the accumulation of pectins in the cell wall of the skin. This is very relevant because calcium is actually involved in mediating crosslinks between pectin molecules, and so this forms a rigid structure in the cell wall. This is thought to be the cause for improved fruit firmness. These results were recently published, so if you'd like to know more about them, you can find about it in this paper. The next step was to understand if calcium was affecting other pathways of the metabolism of the fruits. Grape berries are envisaged as a biochemical factory because they import and accumulate water, minerals, and sugars, and also they synthesize amino acids, organic acids, and phenolics. And so we started by determining several quality parameters of the fruits, including berry weight, reducing sugar content, titratable acidity, Brix, etc. So for the red variety, we confirmed that calcium treatments had no effect on these quality parameters. However, for the white variety, we verified that calcium treatments led to fruits with lower weight and also with a little bit less of sugars, okay, measured by the Brix series. And so this showed us that calcium was delaying the maturation of the fruits of the white variety. The next step was to perform a targeted metabolomic approach to see exactly which metabolites are being affected by calcium. And for this, we established a collaboration with the University of Tours in France. Results showed that for both the red and the white cultivars, there was a clear separation between the metabolic profile of control fruits, here shown in black, and the metabolic profile of calcium-treated fruits, here shown in red. And we actually detected and quantified amino acids, organic acids, and several polyphenolics, including stilbenoids, flavonols, and anthocyanins. We organize the results, we visualize the results in the form of pathways to better understand which routes were being affected by calcium. So when we analyze these pathways, we could conclude that calcium treatments were inhibiting amino acid accumulation. And so this could actually be problematic in the future because many amino acids are substrates for yeast growth. And so this led us to wonder if there could be an impact in fermentation during wine production. Results also showed that calcium treatments increased or stimulated stilbenoid synthesis, and this was also consistent with the upregulation of STS genes encoding stilbene synthase enzyme, which is involved in the branch that leads to stilbenoid accumulation. We observed for both cultivars that there was a significant overaccumulation of epicene and omega viniferin, and for the red cultivar, there was also overaccumulation of resveratrol and epsilon viniferin. This is quite relevant because many of these stilbenoids are phytoalexins produced by the plant to protect itself against fungi infection and fungi toxins. And in particular, resveratrol is very used in the cosmetic industry as an anti-agent metabolite. So this could have a real impact in the consumer dimension. Our results also showed that calcium treatment in the field increased the levels of flavonols in the fruits, and this was consistent with the upregulation of flavonol synthase. These flavonols included camphorol, quercetin, and myristin conjugates. And this is also quite relevant because many of these flavonols have a powerful antioxidant activity in the plant, and their benefits were also shown in many animal cell models. Lastly, our results showed that calcium treatments inhibited the accumulation of anthocyanins, and this was consistent with the downregulation of the expression of UFGT gene, which encodes the enzyme catalyzing the limiting step in anthocyanin biosynthesis. Anthocyanins that we quantified included malvidine, cyanidine, peonidine, and putinidine conjugates in both cultivars. And this is quite relevant because anthocyanins are responsible for the color of red berries and wine, so they directly impact fruit and wine quality, color quality. But in the plant, they are also very important to protect fruits and even leaves from excess sunlight. They can also be used for attracting seed dispersers, among other functions. These results were also recently published, so if you'd like to know more about them, you can find here in these works. Lastly, I'd like to say that our results could be very valuable for the implementation of protocols with calcium sprays for modulating specific properties in grape berries from specific cultivars, such as nutritional value, resistance to abiotic and biotic stresses, and the timing of fruit ripening. I would just like to leave you with a last note in which I like to mention some related works to this work. So we also explore the effect of vineyard calcium sprays on fermentation and wine volatile profile, on grape berry cuticular waxes and surface microbiota, and on post-harvest grape berries, in which we focused mostly on antioxidant activity and pathogen defense pathways. Lastly, we also explore the calcium and hormone crosstalk in the grape berry secondary metabolism. I'd like to thank all our collaborators for their valuable and precious input in this work, and also thank you all and also thank all of you for your attention today.